Bismillahirrahmanirrahim. I am Dr. Zahra Noreen, course instructor of Plant Physiology 1, BOTN 3119. Today's lecture is about last two topics of the chapter photosynthesis. These are C3-C4 intermediates and methods of measurements of photosynthesis. First of all, some studies related to C3-C4 intermediate plants. Kennedy and Lesk reported that in 1974, Maluga verticillata showed anatomical features between C3 and C4 plants with photorespiration lower than in C3 but higher than C4. This species is C3-C4 intermediate during growth stages. Until the third to fourth leaf stage, no bundle sheath appeared and photosynthetic rate was similar to a typical C3 plant. But soon afterwards, the bundle sheath appeared and at 10th to 11th leaf stage, it was seen as C4 plant. In 1998, Rellet et al. Showed that leaf anatomy of C3-C4 intermediate plant Moricandia aroensis was initially C3-like, and number of the mitochondria in bundle sheath cells were the same as those in adjacent mesophyll cells. When the leaf size was between 6 to 12 millimeter, then there was a four-fold increase in the number of mitochondria in the bundle sheath cells. C3 and C4 plants were first described in a report of anatomical variations among plants thought to possess photosynthesis. Among these observations was the first report that the bundle sheath cells of the Maluga verticillata, a plant with C3-like carbon isotope ratios, contained numerous mitochondria and chloroplast arranged in a tight centripetal pattern with mitochondria lining the innermost tangential wall surrounded by chloroplast. A few years later, Kennedy and Lesk reported C4-like compensation points in this otherwise C3-like species. Maluga verticillata became the first reported C3-C4 species. Since that time, a number of important discoveries have been made concerning the mechanism of C3-C4 metabolism, the basis for C3-C4 gas exchange patterns, the nature of genetic control over C3-C4 traits, and the significance of C3-C4 intermediates to the evolution of C4 photosynthesis. Distribution. C3-C4 intermediates have been reported in 25 species from 9 genera representing 6 families. Many of the genera occur in families that also have fully expressed C4 species, although this is not universal, like Moricandia and Parthenium have C3-C4 intermediates but no C4 species. Arguments have been posed to explain C3-C4 intermediates as evolutionary precursors to C4 photosynthesis, although alternative explanations are also possible. Recent studies using formal phylogenetic mapping on the basis of morphological traits and molecular markers have solidified the conclusion that C3-C4 intermediate traits are the evolutionary antecedents to C4 photosynthesis, at least in the genus Flavaria. This is the table showing C3-C4 intermediate species, the name of the species, and their typical habitats. Characteristics of C3-C4 intermediates. Majority of the C3-C4 species have two types of photosynthetic cells. Number one is the mesophyll cells with the chloroplast and some mitochondria. And number two, the bundle sheath cells with the chloroplast and numerous mitochondria usually arranged along the inner wall of the bundle sheath cells. The mitochondria of the mesophyll cells lack glycine decarboxylase, which is an enzyme which is required for the decarboxylation of glycine, and normally it operates in the mitochondria of mesophyll cells in case of C3 plant. But in case of C3C4 intermediate, this enzyme is absent in the mitochondria of mesophyll cells, because of which glycine produced by photorespiration in mesophyll cells must be transported to the bundle sheath cells mitochondria for decarboxylation, as this enzyme is absent in the mesophyll cells. The carbon dioxide released during this reaction is efficiently fixed in bundle sheath chloroplast due to its closeness to mitochondria, which accounts for the lower carbon dioxide compensation points in the C3C4 species. Carbon dioxide compensation point is equal to carbon dioxide produced in respiration over carbon dioxide fixed in photosynthesis. Above the carbon dioxide compensation point, rate of photosynthesis is more, and below it, the rate of respiration is more. These compensation points are actually the values at which the plants cease to provide net photosynthesis. In case of C3 plants, its values ranges from 40 to 60 ppm, while in case of C4 plants, its value is up to 10 ppm. C3-C4 intermediates have ecotypes within a species depending on the geographical distribution. For example, the Maluga verticillata plants from Iowa, Massachusetts, and Mexico are almost C4 plants, while those from Kansas are more similar to C4 plants. These are the carbon dioxide compensation points for three types of plants: C3 plants, C3-C4 intermediates, and C4 plants. As I told you that carbon dioxide compensation points are the values at which net photosynthesis stops. So you can see here its values are higher for C3, lowest for C4, while in case of C3-C4 intermediates, their values are intermediate between C3 and C4. Photosynthesis in C3-C4 intermediates. C3-C4 intermediate species offer an excellent model system for studying the mechanism of photorespiration and the evolutionary aspects of the C4 pathway. The C3-C4 intermediate species seem to have arisen during the process of evolution of C4 plants from the C3 species, and C3 photosynthesis is believed to be the phylogenetic precursor of the C4 pathway. Intermediary characteristics are evolutionary rather than hybrid products. It is evident from the fact in the cruciferi, Moricandia, aroensis, and Panicum milioides are intermediates, but C4 plants have not identified in this family. A partial Kranz anatomy occurs in all the present-day C3-C4 intermediates, and efficient recycling of the photorespiratory carbon dioxide results in a low level of photorespiration. A partial reduction of activities of the photorespiratory enzymes in the intermediate species is evident from limitation in both the extent of glycine production and decarboxylation. Glycine production occurs in one type of the cells, and its decarboxylation enzymes are present in other type of the cells. Glycine production occurs in mesophyll cells, and decarboxylation occurs in the bundle sheath cells. In addition to this, the enzyme compartmentation may be an important factor in reducing the photorespiration. It has been shown recently that bundle sheath mitochondria are enriched with glycine decarboxylase, which would facilitate the efficient refixing of carbon dioxide not only from photorespiration but also from the dark respiration. As I told you in the previous slide that in C3-C4 intermediate plants, production of the glycine and its decarboxylation occurs in two different cells. Production of the glycines occurs in the mesophyll cells, while the decarboxylation of the glycine to serine occurs in the mitochondria of the bundle sheath cells. It is a point to be noted here that in the mitochondria of the mesophyll cells, the glycine decarboxylase enzyme is absent, and it is abundantly present in the mitochondria of bundle sheath cells in C3-C4 intermediate plants. Here, number 1 representing the enzyme Rubisco. GA stands for glycerate, and GI stands for glycolate, and here the 2-PGI stands for the 2-phosphoglycolate, and glycine is represented here by GLY, and serine is represented SCR, and the steric mark indicated the presence of glycine decarboxylase enzyme, which catalyzed the conversion of glycine into serine with the release of one carbon dioxide molecule. And this carbon dioxide molecule will be efficiently utilized by the chloroplast adjacent to the mitochondria in the abundant sheath cells. Hence, this carbon dioxide does not liberated to the environment, rather it is used efficiently by the bundled sheath cells. The efficiency of water use of C3-C4 intermediates is also greater than that of the C3 plants, apparently due to a low carbon dioxide compensation point. Artificial production of C3-C4 intermediates have been attempted by generating hybrids between C3 and C4 species of Atriplex. A few hybrid individuals resemble the parents both anatomically and biochemically, but still were unable to carry out fully integrated photosynthesis. Thus, it appears that a proper compartmentation of photosynthetic reactions seems to be a stringent requirement for C4 photosynthesis, although some studies conducted earlier on hydrilla do not corroborate this conclusion. The inheritance of a complete C4 pathway is rather complex, even though the number of genes involved for each component may not be large. A subsequent study on hybrids between C3 and C4 Atriplex species showed that the inheritance of the biochemistry of the C4 photosynthesis and of Kran's anatomy lie not closely linked, and segregation of a hybrid possessing complete and fully coordinated C4 photosynthesis is a rare event. Thus, conventional breeding methods may not be helpful in incorporating C4 traits into C3 plants. The newer techniques of the plant transformation and genetic engineering are opening up new vistas in this endeavor and are under extensive investigation. Of the C3-C4 intermediates identified so far, those in the genus Flavaria, which appears to be true intermediates, seem to provide the maximum potential for future evolutionary studies of C4 pathway. Interspecific crosses between C3-C4 and C3-C4 intermediate species are quite successful. Further taxonomic, cytogenetic, and genetic studies may be helpful in interpreting the evolutionary data. The second and the last topic of today's lecture is about methods of measurement of photosynthesis. Since photosynthesis is a chemical reaction, its levels are monitored by the rate of occurrence. Changes in the levels of its input and output are used to calculate the photosynthetic rate. Thus, there are various methods to measure photosynthesis. These methods include uptake of carbon dioxide by plant, release of oxygen, dry matter content, and carbohydrate production. First of all, uptake of carbon dioxide by plants. Since carbon dioxide is needed for photosynthesis, measuring how much of it is taken up by the plant gives us information on how much of the photosynthesis is happening. The second technique is the release of oxygen. As we all know that during photosynthesis oxygen is released, so the amount of the oxygen produced during photosynthesis can be measured, and it's an indication of the occurrence of photosynthesis. The next technique is the dry matter content. During the dry matter content is comprised of all the solids in the plant minus the water content, and these solids are all produced as a result of the photosynthesis. So dry matter can be used to measure this process. And the last one is carbohydrate production. As we know that during photosynthesis, carbohydrates are formed. So if we check the production of carbohydrate, we can estimate the rate of photosynthesis in a plant. This is an indirect way of measuring photosynthesis by its product. Parts of the plant can be harvested and dried and weighed at intervals, and the difference in weights gives the increase in the carbohydrates, means sugar, due to photosynthesis. Another technique to measure the rate of photosynthesis is to measure the light-dependent photosynthesis using Hill's reactions. In the first step of photosynthesis, oxygen is produced by chloroplast by separating water molecule using energy from light. Using dichlorophenol indophenol DCPIP as the terminal electron acceptor of the oxygen atoms, Hill's reaction measures the light-dependent phases of the photosynthesis. One of the commercial instruments used for the measurement of the photosynthesis is the infrared gas analyzer, also known as IRGA. Infrared light is absorbed by carbon dioxide. So when infrared light is directed at a plant or leaf of the plant in a closed space or chamber, as shown in the figure, there is less carbon dioxide because the plants have used it in the photosynthesis. So there is more infrared light left unabsorbed in the chamber. The incoming and outgoing carbon dioxide from the leaf chamber is measured by infrared spectroscopy with an internal infrared gas analyzer, and the difference gives us the amount of carbon dioxide from which the rate of the photosynthesis can be calculated. Another technique to measure photosynthesis is chlorophyll fluorescence. Chlorophyll fluorescence is an instrument, a meter is an instrument which based on the principle that when chlorophyll absorbs light, its molecules are raised to an excited state and it returns to its normal state by releasing the energy. Parts of this is used to power photosynthesis. Another portion is emitted as radiation called fluorescence radiation. Since fluorescence radiation is complementary to photosynthesis, it is used to measure it in higher plants as well as algae and in bacteria. In one of my lectures on photosynthesis, I have told you about the fate of the absorbed energy by the photosystems. So, in which I have already explained you that there are three pathways the energy can be utilized from the light, and one is the involvement of the energy into the photochemistry, and this will be utilized for the photosynthesis process. Another way is the dissipation of the light in the form light energy in the form of heat, and another way, the third way, is the dissipation or the wastage, you can say, of the light energy in the form of chlorophyll fluorescence. Of course, the three processes are competing with each other. So if we know the amount of the chlorophyll fluorescence, we can measure the rest of the process with the help of the chlorophyll fluorescence meter. So greater the chlorophyll fluorescence, smaller will be the value of involvement of the light to the photosynthesis process. So this technique is utilized to measure the rate of the photosynthesis, and the yield of the chlorophyll fluorescence emission gives us the valuable information about the quantum efficiency of the photochemistry and heat dissipation. It is important for the plant photosynthesis and ultimately productivity because photochemistry is used to provide energy and reducing power for carbon dioxide dissemination. At room temperature, we assume that the variation in the fluorescence signals arise only from PS2, and we ignore the emission from PS1 largely because the signal does not make a significant contribution below 700 nanometer by PS1. These are the articles from where I have collected the material for the making of this lecture. This is all about the last topic of photosynthesis, and I hope you have learned a lot from the all the lectures which I have delivered on photosynthesis and the rest of the chapters of plant physiology 1. Good luck for the exam. Allah Hafiz.